Assalamu alaikum, everyone. Hope so. You all are enjoying these scientific talks. I, Hafiz Muhammad Amjad, PhD scholar from Government College University, Faisalabad, on behalf of the organizing committee of the Applied Zoological Society of Pakistan and in collaboration with Qaid-e-Azam University, Islamabad, I warmly welcome to all of you on the 4th International Conference on Applied Zoology 2021. I'm the moderator of this 5th session, and in this session, we have a number of researchers from all over the Pakistan who will present their work. And the key areas will be from fisheries, wildlife, biodiversity, and nutrition. Let's start our session. And the first presenter of the session is Dr. Muhammad Mudassar Shehzad from University of Education, Lahore. He will talk about partial replacement of fish meal with locally available black seed for Rahu fingerlings. Please, Dr. Muhammad Mudassar Shehzad.
Bismillahirrahmanirrahim. I am very thankful to the Applied Zoological Society of Pakistan and Qaid-e-Azam University, Islamabad for hosting the 4th International Conference on Applied Zoology 2021. The Applied Zoological Society of Pakistan have already organized 3 successful international conferences in the past few years. The topic on which today we will discuss is the partial replacement of fish meal with locally available black seeds for Rahu fingerlings. Dear viewers, I am Dr. Muhammad Mudassar Shehzad from Department of Zoology, Division of Science and Technology, University of Education, Lahore. There are the certain goals of aquaculture industry. By fulfilling these goals, we can be able to produce a highly proteinaceous fish and can supply to our community. By increasing the production of fish, we can fulfill the demands of increasing human population. That will be helpful to increase the quality of fish. If we talk about the background of problems, currently we are facing the high cost and less supply of fish meal. Due to these issues, we are going to search for alternatives. These alternate protein sources are very beneficial for fish health. By using these alternative protein sources, we can be able to produce a good quality fish feed at low cost. From these alternatives, Nigella sativa, that is called as black seeds, are commonly called as kalwanji in Pakistan, is the basic alternative protein source that is easily available and economically very beneficial. Black seeds have wide range of applications in aquaculture sector. By using the black seeds in fish feed, we can get a healthy fish by increasing its immunological parameters and high performance in terms of growth and digestibility. Here are the results in which, first of all, we will discuss about the growth indices of Rahu fingerlings that were fed on black seeds meal. By this graph, you can observe that the initial weight of all the fingerlings were significantly similar with each other that were stocked in our V-shaped tanks before the starting of our trial. But if we talk about the final weight of fingerlings, we can observe that the fish fingerlings that were fed on 10% of black seed meal were showing the highest weight gain as compared to the fish that were fed on control and other test diets. But the fish that were fed on 10% of black seed meal was significantly similar with the weight gain of fish that were fed on 20% of black seed meal. Similarly, best FCR and highest SGR were also observed in the fish fingerlings that were fed on 10% of black seed meal, following the fish that were fed on 20% of black seed meal. If we talk about the nutrient digestibility, we can also observe that the fish that were fed on 20% of black seed meal showing the highest digestibility of crude fat, crude protein, and maximum gross energy as compared to the fingerlings that were fed on control and other test diets. When we talk about the hematological parameters of Labiorhita fingerlings, we have also observed that the fingerlings that were fed on test diet number 3 having 20% of black seed meal showing the best values in terms of RBCs, in terms of hematocrit, in terms of hemoglobin, and in terms of platelets. Whereas the fish fingerlings that were fed on control and the test diet number 6 supplemented with 50% of black seed meal were showing the least values of RBCs, HB, platelet, etc. At the end, we have analyzed the mineral absorption in fingerlings that were fed on black seed meal. By this table, you can easily observe that the blue color is showing the highest values of mineral absorption in fingerlings. Here again, you can see that the fish fingerlings that were fed on 20% of black seed meal supplementation level showing the best absorption values in terms of potassium, calcium, sodium, and phosphorus. Whereas the lowest values of all of these minerals were found in the fish that were fed on controlled diet as well as the fish that were fed on 50% of replacement fish meal replacement level. Whereas if we talk about the iron was maximally absorbed in fingerlings that were fed on 30% of black seed meal, that is called as test diet number 4. On the other hand, if we talk about the copper absorption, copper was maximally absorbed in the fingerlings that were fed on test diet number 5. And the magnesium was maximally absorbed in the fingerlings that were fed on controlled diet. If we talk about the conclusion of our study, we can conclude that the fish that were fed on test diet number 3 having 20% of replacement with fish meal of black seed meal having the best values in terms of growth, hematology, nutrient digestibility, and mineral absorption. By this study, we can give an approach towards the alternative protein sources, such as black seed meal. By using black seed meal at 20% of its replacement, we can get a healthy fish for our community and with a very low cost. I am very thankful for your attention, please. If you have any question, you may ask, please. Thank you.
Thank you very much, Dr. Muhammad Mudassir Shehzad. It was really informative talk. The next presenter of the session is Ms. Sahar Saleem from University of Agriculture, Faisalabad. She will talk about a comparative study on the accumulation of copper and cadmium in different tissues of labiorrheta. Please, Ms. Sahar Saleem.
First of all, I would like to thank the organization committee of ICAZ-2021 for providing us this great opportunity to share our ideas on this platform. My name is Sahar Saleem, and I have done MPhil in zoology from University of Agriculture, Faisalabad. Today, my topic of our presentation is a comparative study on the accumulation of copper and cadmium in different tissues: gills, liver, kidney, spleen, muscle of labiorrheta. As introduction, fish are commonly used to determine the health of marine environment because contaminants raised in the food web. The fish are responsible for toxic impacts on fish death in the marine ecosystem. Heavy metal contamination may have devastating effects on ecological balance of the recipient environment and diversity of aquatic organisms. The accumulation of heavy metals in fish mainly dependent on metals concentration in water and exposure period, although some physical factors. Metals are non-biodegradable and are considered as major environmental pollutants, causing cytotoxic, carcinogenic effects in animals. Cadmium is a genotoxic metal to aquatic organism by inducing the generation of oxidative stress and immune toxicity. Cadmium and copper are well known to induce hematotoxicity in fish, resulting in immune suppression in contaminated water. However, copper is an important essential element in various metabolic processes, neurotransmitter function. The objective of this study was a comparative study on the accumulation of copper and cadmium. These are the heavy metals that are present in water. Effects of copper and cadmium on gills, liver, spleen, kidney, and muscle tissues of labiorrheta. Material methods that are used in this research to determination of physical chemical parameters, water temperature, dissolved oxygen were measured by electronic meter, while pH were measured by digital meter in lab. However, total ammonia, hardness, calcium, magnesium, carbon dioxide, and measured by APHA 1998 method. Determination of metal in fish organs, concentration of metal in fish tissues were calculated from fresh mass of each organ, resulted were given as wet weight. Cadmium and copper accumulation were also calculated after the exposure of 15, 20, 30, 45, 60, 75, and 90 days interval. And determination of exposure of metals like cadmium and copper through atomic absorption spectrophotometer. Here are the results. In this graph, level of copper was measured in kidney by falling order, in which spleen shows the highest rate of exposure of this metal, then gills, then comes these muscles, then liver, and kidney shows the lowest effect of this metal in this organ. In this case of cadmium, the level of cadmium was measured in kidney by falling order. In fish, kidney also showed the effect of cadmium on in kidney. Then muscles, then spleen, gills, and then liver. Liver shows the lowest effect of this metal. The conclusion of my search, 90 days experiment was conducted at Fisheries Research Farm, Department of Zoology, Wildlife and Fisheries, University of Agriculture, Faisalabad. In tissue of fish from the organized set, like kidney, muscle, spleen, copper was 3.1 to 38.1 microgram per gram, as concentration of cadmium were much lower, at 0.1 to 3.6 microgram per gram. The accumulation patterns of both metals were diverse in the dissimilar organs of fish. Means both metals showed different effect on different organs of fish. High effect of copper was seen in spleen, while in case of cadmium, kidney is affected by this metal. Physicochemical parameters show significant correlation with different concentration of copper and cadmium. These are the conclusions that was in my research. Thank you for your attention.
Thank you very much, Ms. Sahar Saleem. It was really wonderful talk. The next presenter of the session is Mr. Bilal Ahmed from Government College University, Faisalabad. He will talk about efficacy of citric acid and phytase treated cottonseed meal-based diet in Ketla fingerlings to improve mineral digestibility. Please, Mr. Bilal Ahmed.
You start with the name of Allah Almighty. My name is Bilal Ahmed, PhD scholar, Department of Zoology, Government College University, Faisalabad. The topic of my presentation: efficacy of citric acid and phytase treated cottonseed meal-based diet in Ketla fingerlings to improve mineral digestibility. The worldwide acquisition of aquatic food is increasing nowadays because of increasing human population. The availability of aquafeed products lead to the expansion of aquaculture sector. The reasons behind the development of aquaculture sector are that it is the source of economical feed which have better growth ability in short period of time, and to achieve low budget farming, the suitable way is the formulation of artificial feeds from available natural products such as plant stock. The source of protein is regarded as an important ingredient in fish meal for feed formulation, but its less availability and its cost may limit its use in fish nutrition. The main ingredient that I have used in my research, cottonseed meal, has been used as feed ingredient in terrestrial and aquatic animal feeds as an excellent source of protein. Cottonseed meal has protein content up to 44% after soybean meal and canola meal. It is the third largest oilseed meal in world. In order to decrease the utilization of fish meal, proteins from animals and plant sources have been used. However, the poor digestibility and palatability of these proteins and also imbalanced amino acid profile, which lead to severely restricted their use in aquafeed, is due to the presence of anti-nutritional factors. This slide shows cottonseed and cottonseed meal. The first supplement that I have used in my research is phytase. Phytate from complexes with divalent and trivalent ketynes, as phytate is negatively charged ion, thus lead to adverse effect on mineral absorptions. Microbial phytase is an enzyme that makes phosphorus availability to fish and enhances its absorptions, releasing phosphate group from phytate. The activity of phytase depends on pH and it's highly active at low pH of gut. The production of acid lead to lower the pH of gut in carnivore fish, but on the other hand, monogastric fishes lack this ability. The next supplement that I have used is citric acid. Citric acid has become the world's most usable organic acid. The inclusion of organic acids such as citric acid in fish diet lowers the pH of digestive tract, and the activity of phytase becomes maximum by using of organic acid. It was confirmed that citric acid is being successfully utilized due to its ability to hydrolyze the phytate complex and lead to enhance the nutrients absorption as well as resistance against diseases. The fish I have used, Catla. is ordinarily known as thala, extensively cultured herbivore fish that use mesh feed from the water surface as surface feeding fish. Among other major carp plains like Ligurita and Surhinus meridella used in Pakistan, Catla is generally used as a part of polyculture production system. It is one of the fastest growing fish among other carps due to its highly nutritious values and also good taste quality and thus has valuable market demand. Materials and methods, fish and experimental conditions. For a 90 days trial, Catla fingerlings were purchased from government issued hatchery, Faisalabad, treated with 5 gram per liter NaCl solution before stopping, brings them in fish nutrition lab, Department of Zoology, Government College University, Faisalabad. Here, the fingerling acclimatized for 2 weeks, continuous irrigations and water quality parameters were checked. Feed formulation were done by grinding and mixing of all ingredients that represents in table 1. Table 1 represents the different composition of ingredients. I have made 16 test diets in 4 groups. Fish meal protein replacement 4 groups: 0%, 25%, 50%, and 75%. Feeding protocol and sample selection. Feeding protocols 5% at their live fat were given to the fingerlings. After feeding, washing of tanks, feces selection by the subsequently opening of valve 1 and valve 2. The obtained feces dried and analyzed. This slide shows feeding plate and mesh size. This slide shows about the valve 1 and valve 2 used for fecal collection. The mineral analysis was done by different instruments: UV, VIS, spectrophotometer, atomic absorption, flame photometer, apparent digestibility coefficients calculated by standard formulae, statistical analysis. Data of minerals digestibilities was fitted to 3-way ANOVA. Results. Table 2 represents percentage of minerals that I have used in test diets of Catla fingerlings, fed on cottonseed meal-based diet supplemented with citric acid and phytase. Table 3 shows the composition of minerals present in feces that obtained from Catla fingerlings fed on cottonseed meal-based diets. This table shows 4 replacement levels and maximum minerals in feces present in control diet TD1 and minimum composition of minerals in feces or obtained test diet TD12 that have inclusion of both phytase and citric acid, while on the other hand, the test diet 1 have no phytase, no citric acid. Table 4 shows apparent minerals digestibility of Catla fingerlings fed on cottonseed meal-based diets. Here we can also see that at 50% replacement level, test diet 12 that have both phytase and citric acid have maximum values of minerals present in Catla fingerlings as compared to that of 0% protein replacement level. Test diet 1, control diet having no inclusion of phytase and citric acid have minimum values of minerals present in Catla fingerlings. Based upon the finding, it is stated that test diet 12, which contains up to 50% cottonseed meal protein, 2.5% citric acid, and phytase, 750 phytase, is the most effective diet for increasing mineral digestibility in Catla fingerlings. This slide shows the references. Thank you for your attention.
Thank you very much, Mr. Bilal. It was really wonderful talk. The next presenter of our session is Dr. Danish Riaz from University of Education, Faisalabad. He will talk about efficacy of probiotic supplementation on growth and nutrient digestibility of Cirrhinus merigella fingerlings fed Moringa oleifera oil seeds by products-based diet. Please, Dr. Danish Riaz.
First of all, thanks to the Applied Zoological Society of Pakistan and Hyderabad University of Pakistan for providing the dynamic opportunity for the international collaboration in the form of the International Conference on Applied Zoology 2021. So this is a good platform for exploring the effective solution towards the animal welfare and sustainability. I'm Dr. Danish Riaz, IBFE Fellowship at Department of Zoology, Ministry of Education for Kasawasa campus. The first topic is the efficacy of probiotic supplementations, supplementation of the growth, nutrient digestibility of the Aeronoconas medicus fingerling fed the Moringa oleifera oil seed byproduct-based diet. Agriculture is one of the important industries in the world. This is increasing from 109 will be 109 million tons in 2030, as compared to the 82.1 million tons in 2018. Agriculture is the fastest growing food producing industry in the world. Government aquaculture is more than any other food production sector. However, the economic factors such as cost of fish meal is limited towards development. Fish meal is a healthy source of nutrients because it contains the essential amino acids, fatty acids, highly palatable, and contain the high digestible protein. Some problems with nutrition due to the rising demand of the fish meal, and third, highly cost effective, and fourth one is the static level of production of the fish meal. Plant byproducts, these are products, these are the so good source of protein and energy, environmental friendly, and cost effective. Moringa oleifera is one of the best source because it's a good source of sulfur-containing amino acids such as methionine, cysteine, tryptophan. It also contains the fats and minerals. Probiotics are the microorganism and the substance that contribute to the intestinal microbial balance and microbial cells and distributed in a certain way, which improves the gastrointestinal tract remain alive with the aims of improving the health and growth. These are the probiotics which are helpful for the growth as a growth promoter and nutrient digestible. Probiotics are the rich source of nutritional ingredients such as protein, fat, fatty acids, vitamins, proteinoids, such as potential antioxidant. It could be a good food adapter for aquaculture. These are the multi-strain probiotics which contain the 9 strains which are used in our research work. Research is needed to discover the suitable and the least cost-effective plant source to discover the problem of costly fish meal to minimize the aquaculture pollution caused by the minimal nutrients escaped. Oilseed byproduct-based diet supplemented with the probiotics increase the nutrient digestibility of the Aeronoconas medicus, resulting in a high growth. To determine the effect of probiotics on the growth and nutrient digestibility, to determine the exact and accurate level of the probiotic concentration. These are the fish in experimental conditions such as procurement, implementations, and control of water quality parameters. Moving on, if you have base diets is further divided into 7 diets: 0, 0.5, 1, 1.5, 2, 2.5, and 3 gram per kg of probiotics level. Feed ingredients and experimental diets. These are the ingredient composition of the test diets, chemical composition of the ingredients. This is the feeding protocols. Triplicate tanks was used, and each tank contains the 15 finger lengths. These are the experimental design. These are the chemical analysis of disease. Growth parameter study by using the standard formulas: SCR, specific growth rate, FCR, feed conversion rate, and weight gain. Definition of new digestibility. Post-stat computing methods used for the one and over different tests and results. Growth performance of the coronavirus neuroticals fed the level of probiotic protection supplemented the amino in their diet. It is noted that the 2.5 gram per kg probiotic concentration is the optimum level for the maximum growth rate in terms of growth gain, weight gain, grams, growth, weight gain, percentage, FCR and FCR. At this level, the FCR is the lowest values, which is the optimum for the maximum growth rate, and at the 3 gram per kg of probiotics is just drastically reduced. So the 2.5 gram per kg probiotic concentration is the optimum and significantly different from the control diet, which is without the probiotics nutrient digestibility. Analysis of composition of the episode-based diet and nutrient segment. There is a nutrient digestibility, which is maximum at 2.5 and 2.5 gram per kg concentration. Supplementation of the probiotics and nutrients in the fishes at 2.5 is the optimum range. Research provided sufficient evidences that probiotic supplementation that create the phytate complex present in the amazon-based diet improves the overall growth performance of coronavirus neuroticus. From this finding, it was concluded that the supplementation of probiotics play a major role in the growth performance and nutrient digestibility of the coronavirus neuroticus finger leg as compared to the control diet. These are the experimental. It's a both helpful and probiotics byproduct based diet in the high quality and affordability for the fish farmer at the small scale in Pakistan. That the short outcome. These combinate that effect of probiotics on the effect on genetic makeup of the coronavirus should be checked. That was analyzed. There were not analyzed in present studies. It is commented that commercially important and cost-effective probiotics in the fish feed for the local farmer. It is also commented that the further study should be considered on the other plants meals, but not observed in the studies. So the further research is recommended for the exact to analyze the exact level of level for the maximum fish growth. It might be possible. The research was these are the references. Thank you for your attention.
Thank you very much, Dr. Danish Riaz. It was really a wonderful talk. The next presenter of our session is Ms. Ikra Yunus from Kohart University of Science and Technology. She will talk about effect of neem leaf supplemented diets on growth and hematological parameters of grass carp. Please, Ms. Ikra Yunus.
My name is Ikra Yunus, student of MPhil Zoology, Department of Zoology, Kohart University of Science and Technology. The topic of my presentation is effect of neem leaf supplemented diets on growth and hematological parameters of grass carp. Presentation outline, introduction, objectives, material and methods, results, conclusion, recommendation, acknowledgement, references. Introduction. The neem tree, botanically referred to as Astracta indica, belongs to the family Citric. It is a fast-growing evergreen tree that may grow up to 20 meters in height. It is one of the most useful traditional medicinal plants. Almost every part of the neem tree, including seeds, leaves, roots, bark, trunk, and branches, have multiple uses, but most of the pharmacological activities have been described especially for neem leaves. The species which I was used and now researched is grass carp. Grass carp is a herbivore. Grass carp scientific name is Stenoceranodon ivila. Grass carp is one of the essential freshwater aquaculture species with the top production among all species around the world. Growth in grass carp is a function of age, size, abiotic factors such as density, nutrition, temperature, and oxygen. Grass carp requires cellulose and protein in their diet, with protein being especially important for optimal growth and young fishes. Objective. The first one objective is to evaluate the potential effect of neem leaf supplemented diet on growth performance in grass carp, and the second one objective is to determine the effect of neem leaf supplemented diet on hematological parameters of grass carp. Materials and methods. The first one is sample collection. The second one is leaf collection. The third one is diet preparation. The fourth one is growth analysis. And the fifth one is hematological analysis. And the sixth one is statistical analysis. Data. Here we are seeing two tables of result. The first one table shows growth of each fish and the aquarium before supplemented diet, before exposed to supplemented diet. The second one table shows growth of each fish after exposed to supplemented diet. Here we see clear difference between these two tables, but for further clarification, I have got two graphs for these two tables. The first one graph is for the first one table, and the second one graph is for the second one table. And these are positive effect of supplemented diet on the growth of fishes in these two graphs. This one is the second graph for second table. Here we can see clear difference between the growth. The concentration of neem in the experimental diet is the 2 gram in T1 and 5 gram in T2 and 10 gram in T3. Results. Hematological analysis. Red blood cells, lymphocytes, eosinophils, monocytes, basophils. These are all the hematological parameters. First one is the red graphical representation of hematological parameters. Red blood cells, red blood cells. Red blood cell count of blood of grass carp species decreases as the neem concentration in the feed increases. The second graph shows that the lymphocytes count of the blood in grass carp is increases as the mean concentration in the feed of fishes increases. The third one is hemoglobin parameters, hematological parameters. Hemoglobin counts of blood in the grass carp is decreases as the mean concentration in the feed increases. Similarly, the fourth one, eosinophil and monocytes. These two are similar as the mean concentration in the feed of the fishes increases. These both parameters are also increases. I didn't draw graph for the basophils because the mean concentration, mean diet have no effect on the basophils. Conclusion: neem leaf supplemented diet are more effective compared to normal commercial diet of fish. The second one is neem leaf supplemented diet acts as a growth or immune enhancer. The diet prepared from neem leaf are cheapest as compared to commercial fish diet. Neem leaf inclusion in feed has a remarkable positive effect in the improved general growth performance. Recommendation: neem leaf supplemented diet of fish should be prepared with high concentration of neem. Its negative effect at high concentration level should be recorded. Improper technology should be standardized to alleviate these effects. Acknowledgement: I am very thankful to my supervisor, Dr. Farmanullah Dagar, and my co-supervisor, Mr. Salman Ahmed for their guidance and support. Thanks to all faculty members, my friends, my family, and also my lab fellows. These are references. Thank you for your attention.
Thank you very much, Ms. Ikra Yunus. It was really educative talk. The next presenter of our session is Mr. Muhammad Zubairul Hassan Arsalan. He will talk about synergistic effects of citric acid and phytase supplemented canola meal based diet on growth performance and nutrient digestibility of Cyprinus carpio fingerlings. Please, Mr. Muhammad Zubairul Hassan.
My name is Muhammad Bilal Ahmed, Department of Zoology, Government College University, Faisalabad. My research topic is effect of the phytase and citric acid supplementation on growth performance and nutrient digestibility of Cyprinus carpio fingerling fed on canola meal based diet. Aquaculture development more efficiently than an other food production sector. However, economic factors such as cost of fish feed are limiting its development. Aquaculture feed industry mainly depend on the use of fish meal due to the high nutrition and palatability values. Fish meal. Fish meal rich in essential amino acid and fatty acid required for optimum fish growth. However, its availability has alarmingly reduced in recent years due to over exploitation, natural fishing ground resulting in tremendous increase in its price. Feed primarily accounts for 50 to 60% of total cost in the fish farming. The main object for most fish farmers is to produce high quality fish feed at low cost. Increasing fish meal price motivated the scientists to identify the cost effective alternative of fish meat. Plant byproducts. Plant byproducts are the promising source of protein and energy. Canola meal is the valuable protein source for use in the animal feed, and it's contain 32 to 39% protein contents. Its amino acid profile is similar to that of herring meal protein as superior to soybean meal. Canola meal is being used up to 20% inclusion levels in fish diets, but it decreased to further replacement. Fish meal is in the diet due to the limited world supplies and the growing demand for these highly valuable species. This is a view of canola meal. Canola meal Canola meal in fish diets. Canola meal is the well established as a feed ingredient in salmon and trout diet, where it has been routinely fed on over 20 years. Canola meal is the use at up to the 20% ingredient level in salmon diet, but it is decreased to the further displace fish meal in diet due to limited bird supplies and growing demand for these highly valuable species. This is the structure of phytate. Phytate supplementation, phytate breakdown and convert into the phytase. Role in the citric acid. Decrease to the pH of digestive system, stimulate the feed consumption that will result to the better absorption of nutrients, better growth performance. This is the view of Saprinus carpio Saprinus carpio fish. Saprinus carpio is the commonly known as gulfam, bottom feeder, high consumer choice, widely cultivated under polyculture system. This is the material message used. Fish and experimental condition. Fingerlings were purchased government fishes hatchery. Fingerlings were treated with 5 gram per liter NaCl solution. Acclimatization for 2 weeks. Continuous aeration determined. Water temperature and pH also determined. This is the composition of feed ingredients. Feed formulation. Firstly, the feed ingredient analyzed, grinding, and electrical mixing. Designer view or fish tanks. This is the collection tube, wall 1 and wall 2. Collection of the nutrient digestibility for these formulae. The chemical analysis of fish and fishes. Firstly, the samples was grinded with the help of pestle and mortar, crude protein, crude fat, and ash and gross energy by deducted of the bump calorimeter. Statistics analysis. Collectively, nutrient digestibility and data was experimental diet was the subject of the three-way NOVA of the variance. The difference among treatment was the compared and considered significantly. The co-stat package use the result. The result is the present where we concluded the maximum digestibility on the nutrients canola meal with diet was the observed on the diet T12, which have 25% citric acid and 750 FTU phytase enzyme. The secondly, best result value was the obtained in the T11 diet, which have 0% citric acid and 750 FTU per kg phytase. And the lowest the lowest digestibility show in the T1. This is the values percentage tables in the feces in the table. Conclusion. The conclusion in the recent studies provide evidence that the citric acid and phytase supplementation canola meal based diet fed down the Saprinus carpio fingerling in the nutrient digestibility and without the negative effect of growth and carcass composition at the 2.5% citric acid and 750 FTU per kg level. It also reduced the discharge of the nutrients into the water, resulting in environment friendly and cost effective fish culture. Thank you.
Thank you very much, Mr. Zubair. It was really a nice talk. The next presenter of our session is Ms. Nimra Abbas from Bahauddin Zakir University, Multan. She will talk about hematological studies of freshwater fish, Labiorheta exposed to engineered zinc oxide nanoparticles. Please, Ms. Nimra Abbas.
Institute of Biotechnology and from Boston University, Bhutan. And the topic of presentation is hematological studies of freshwater fish that are exposed to engineered zinc oxide nanoparticles. First of all, I will tell you introduction of this research work. The freshwater ecosystem are biological ecosystem in which biologic sections show their growth and operations. And from the human activities, metals and other inorganic and organic particles are also gathered in this ecosystem. The most common pollutants which are very dangerous for the fishes and other aquatic bodies are cadmium, chromium, copper, nickel, and lead. The heavy particles start gathering in the fish bodies, that means they become the part of food chain and food web. All the product of heavy particles are poisons for the aquatic bodies. And against of these metals, the small amount of nanoparticles are also becoming the part of freshwater ecosystem. And these nanoparticles are inorganic particles, and all of these are toxic and dangerous particles, which reach to the aquatic ecosystem and they start depositing in the living bodies as well as in the water. The mode of action of these nanoparticles are that when they enter the water, they interact with the cells and start producing the reactive oxygen secretions named ROS. And these ROS cause the antioxidant defense. Or as a result of this antioxidant defense, the oxidative stress takes place, including in this oxidative stress, including the damage to the DNA, protein, and lipid take place. And the result of this oxidative stress is that inflammation in the body occur and make the cell death as a process and a process. And the second, zinc oxide nanoparticle also show the same procedure. Zinc oxide enter into the nanoparticle, zinc oxide enter in the body and cause the ROS generation and damage the micromolecules and organelles, and other process occur, and sometimes cytotoxic against the cell. Then hematological parameters which are used to measure and examine the fish health without eating that animal. These parameters such as hematocrit, hemoglobin, red blood cells, white blood cells, MCH, MCB, and MCC are also be used as an indicator of disease and stress used in the case of aquatic ecosystem that are used to monitor the environmental toxicity. When the small amount of zinc oxide nanoparticle exposed to the fish, the level of erythrocyte in red blood cells, hemoglobin, and hematocyte HCT low down as compared to the control, this result is shown because of hemolytic reaction in the body. The hemolytic reaction is that when the cells that form red blood cells decreased and the oxygen capacity also decreased. And small amount of zinc oxide nanoparticle increase the white blood cells and platelets because of defense mechanism against of these heavy particles. And some researchers also show that the engineered nanoparticles are very toxic and cause the damage in the nucleus function, resulting in fighting ability of these cells against the viral and bacterial function decreased. And material methods. The material methods include that first of all fish acclimatization. For the fish acclimatization, we put the fish in the tanks of steel for a time of 2 weeks at the watery condition. Fish were fed with a supplementary diet during the acclimatization. After this acclimatization, the fishes were shifted to the glass aquarium of 50-liter water capacity. And an experimental trial took place. And for this experiment, the glass aquarium was used, which having the water capacity of 50 liters, zinc oxide nanoparticles added to maintain the suitable concentration. After this, the continuous oxygen supply was maintained with a capillary system and also with the electronic oxygen pumps. For the experiment, the fishes were fed with the commercial supplementary diet continuously. And methodology for the study of hematological parameters is that blood samples were collected after each 5 days. The trial was took place for the 15 days, and samples were taken after 5, 10, and 15 days for the study of effect of zinc oxide nanoparticles on rat fishes. Fish size is the 12 to 14 centimeters, and its weight is 10 to 30 grams. And then fish was anesthetized, then under the sterile condition, blood samples were taken by puncturing with a syringe of 2.5 ml. And this syringe was put on the posterior wing in the biological methods. And the method which I used for that study is the hematocrit value was studied with the help of micro hematocrit capillary tube, and red blood cell and white blood cells counting take place with the help of Hill cytometer. And hemoglobin concentration and means in hemoglobin concentration was studied with the help of cyan methioglobin, and its formula is also given. And means in hemoglobin and means in volume was studied with the help of that formulas which are mentioned by the Desi and Levis in 2001. And the next is results. These graphs shown that in the first the WBC's concentration increase will increase the duration and exposure of zinc oxide nanoparticles. The first is control, and second graph is the dose 1, and third one is the dose 2. And, the MCSC also increase the concentration of zinc oxide nanoparticles, and the duration its concentration also increase with the duration of exposure zinc oxide nanoparticles. And mean cell volume also be increased and increase the zinc oxide nanoparticle concentration because defense mechanism and or inflammation. MCSC also be increased, and its concentration is a little bit increased because its concentration does not depend upon the zinc oxide nanoparticles. And the next is red blood cells. Red blood cells concentration also be increased, also be decreased within the cell nanoparticle due to the hemolytic reactions. And hemoglobin concentration is also be decreased, and hematocrit concentration is also be decreased because they are directly proportional to the zinc oxide nanoparticle. When the zinc oxide nanoparticle increase, there is exposure increase, duration increased, then hemolytic reaction also be increased, and its concentration decreased. Thank you for your attention.
सुनो इसे मोबाइल पे
Thank you very much, Ms. Nimra Abbas. It was really a wonderful talk. The next presenter of this session is Mr. Ghaniur Rahman from Kohag University of Science and Technology. He will present his work on the effects of chlorpyrifos on hematological and biochemical parameters of tenophrenton idella. Please, Mr. Ghaniur Rahman.
Bismillahirrahmanirrahim. I am Ghaniur Rahman from Department of Zoology, Kohag University of Science and Technology. I have done my MPhil work on the topic of effects of chlorpyrifos on hematological and biochemical parameters of grass carp under the supervision of Dr. Farmanullah Dawad and Dr. Saeed Ishtiaq Anjam. The outline of my presentation is introduction, objectives, material and methods, results, conclusion, recommendations, and references. Introduction. Contamination is a widespread source of environmental pollution and a matter of concern all over the world. Pakistan is an agriculture-based country. Chlorpyrifos is a broad-spectrum insecticide currently used in more than 100 countries worldwide. Chlorpyrifos run off into water bodies through different ways. Chlorpyrifos is relatively inexpensive and highly efficient. However, its excellent chemical stability and environmental persistence pose potential threats to aquatic life and to the environment. Chlorpyrifos toxicity to fish. Chlorpyrifos is highly toxic to aquatic organisms. It affects fish behavior and physiology. It also affects growth and reproduction and induced histopathological alterations. It also adversely affects immune response and blood indices. It also affects enzymatic and hormonal activities of fish. It also causes impairment of hepatic metabolism and genotoxic damage. Grass carp which is also known as Ctenophrygmodon edela, as a species of freshwater sapronid fish, which is native to China. Grass carp was introduced in Pakistan from China in 1964. Since then, grass carp became a popular market fish in Pakistan due to its meat quality and high economic value. Fish is an excellent bioindicator for any contamination. Objectives of my research is to analyze the potential effects of chlorpyrifos on biochemical indices of grass carp and to investigate the potential effects of chlorpyrifos on hematological parameters of grass carp. Material and methods: collection and acclimatization of grass carp, chlorpyrifos dilution, acute toxicity experiment, LC50-96 source determination, hematological study, and blood biochemical study. These are some patches taken during experimental work: chlorpyrifos dilutions, acute toxicity experiment, blood collection, blood preservation, and then centrifugation, then biochemical analysis. Hematological analysis. Hematological parameters included red blood cells, white blood cells, hemoglobin, hematocrit, and mean corpuscular volume, mean corpuscular hemoglobin, mean corpuscular hemoglobin concentrations. Biochemical analysis including glucose concentration, albumin concentration, triglyceride concentration, and cholesterol level of blood, and then total protein content of the blood. Results: behavioral response of Ctenophrygmodon edela exposed to different concentration of chlorpyrifos for 96 hours at 25 degrees centigrade. They show different abnormal behavior during different concentrated groups. Fishes were divided into 4 groups. They show different behaviors like fast swimming, loss of equilibrium and balance, gulping, pale body color, excess amount of mucus secretions. Able to show acute toxicity test of chlorpyrifos exposed to chlorpyrifos. On the left side, the different concentration of chlorpyrifos, and we have divided the fishes into 5 groups. First one was treated, was controlled, while the remaining 4 were treated groups. Treated one was the group 1 was treated with 50 microliter, while the treated 2 was treated with 100 microliter concentration, while the third one was treated with 150 microliter. The fourth one was treated with 200 microliter chlorpyrifos concentration. Determination of LC50 values of chlorpyrifos and Ctenophrygmodon edela for 96 hours at 25 degrees centigrade. This is the table in which you can see the number of dead fishes and mean death, then mean death and concentration difference. Then according to the formula, we have found the LC50 of chlorpyrifos for grass carp, which is 165 microliter per liter. This slide shows the graphical representation of hematological analysis. the red blood cell content in control group and chlorpyrifos treated group 1, chlorpyrifos treated group 2, and chlorpyrifos treated group 3, and chlorpyrifos treated group 4. The red blood cell content of blood of grass carp exposed to different concentration of chlorpyrifos. The red blood cell content of blood showed decrease with increase in the concentration of chlorpyrifos. This slide is about the white blood cell content of grass carp exposed to different concentration of chlorpyrifos. The white blood cell content show increase with increase in concentration. As you can see, in control group and chlorpyrifos treated group 1, in chlorpyrifos treated group 2, 3, and 4. They show white blood cell were increased and chlorpyrifos treated group 4 with increase in chlorpyrifos concentrations. This slide is about the hemoglobin concentration of grass carp exposed to different concentration of chlorpyrifos. The hemoglobin content show decrease and blood with increase in chlorpyrifos concentration. As you can see, the hemoglobin concentration were decreased with increase and the concentration of chlorpyrifos. This is the graphical representation of biochemical analysis. This slide is about the glucose concentration, glucose level, glucose content of blood of grass carp exposed to different concentration of chlorpyrifos. Glucose level were increased with increase in chlorpyrifos concentration. This slide is about the albumin level of grass carp exposed to different concentration of chlorpyrifos. The albumin content also showed decrease with increase in chlorpyrifos concentration. As you can see, at 24 hours, at 48 hours, at 72 hours, at 96 hours, the albumin concentration showed decrease in blood with increased concentration of chlorpyrifos. Conclusion: we assume that chlorpyrifos has strong effect on the blood biochemical and hematological parameters of fish tested. We can conclude that chlorpyrifos is highly toxic to fish. These parameters can be used as effective guidelines for the toxic level indices for farm grass carp fish. Moreover, pesticide used on farmlands eliminate other non-target organisms and also pose a risk to human health. Recommendations: the use of chlorpyrifos insecticide must be at a minimum level. Indiscriminate use of pesticides should be avoided, and future effect of chlorpyrifos insecticides on grass carp can be studied on genetic level, or then it can also call it genotoxicity. These are references. Acknowledgement: I am very thankful to my supervisor, Dr. Farmanullah Dawad, and co-supervisor, Dr. Sayyid Ishtiaq Anjam, for their guidance and support. Thanks to all faculty members, my friends and lab fellows. Thank you so much.
Thank you very much, Mr. Ghaniyur Rahman. It was really a nice talk. The next presenter of our session is Ms. Sadia Tabassum from Government College, University, Faisalabad. She will talk about fish meal replacement with Moringa oleifera leaf meal and its impacts on growth performance, nutrients absorption, and minerals availability in Labiorhita fingerlings. Please, Ms. Sadia Tabassum.
Start with the name of Allah, which is most beneficent, most merciful. This is the 4th International Conference on Applied Zoology 2021. My name is Sadia Tabassum. I'm from the Department of Zoology, TC University of Islamabad. The research topic is fish meal replacement with Moringa oleifera leaf meal and its impact on growth performance, nutrients absorption, and minerals availability in Labiorhita fingerlings. Aquaculture. Aquaculture is the fastest growing food producing industry. It is developing more efficiently than any other food production sectors. It has a potential for meeting the demand of quality protein, but have raised several issues that need to be addressed. One of these issues is development of fish feed from high quality, inexpensive sources, methods for making the feed free from anti-nutritional factors, cost of fish feed. These are the economic factors that are limiting its development. Fish meal is the major source of protein and essential nutrients in fish feed. However, increasing demand, unstable supply, and high price of the fish meal with the expansion of aquaculture make it necessary to search for alternative protein sources. Feed primarily accounts for 50 to 60% of total cost in fish farming. The main objective for most fish farmers is to produce high quality fish feed at low cost. Increasing fish meal prices motivated the scientists to identify cost effective alternatives of fish meal. Plant byproducts, which can be used as a replacement of fish meal. Plant byproducts are the most promising sources of protein and energy for aquafeed. It is biodegradable. It has cost-effective. It is environment-friendly fish feeds. Moringa oleifera leaf meal is a promising protein source at low levels. It is important sources of vitamins and minerals. It is rich in essential amino acids, carotenoids, ascorbic acid, and minerals, especially iron. Due to favorable amino acid profile and wide availability, Moringa oleifera leaf meal to make aquaculture production cost effective. It has high amount of other minerals such as calcium, copper, zinc, potassium, phosphorus, magnesium, and sodium. It contains crude protein 25% to 32%. It is a miracle tree, has an impressive range of medicinal uses. It has highly digestible, due to a high proportion of pepsin-soluble nitrogen and low proportion of insoluble protein. Labiorhita is the mature Indian carp. It is commonly known as Rohu, cultured in Pakistan, Column feeder. It has high commercial value due to consumers' price. It is widely cultivated under polyculture system. Materials and methods. Fish and experimental condition. Labiorhita fingerlings was were purchased from government fish seed hatchery, Satyana of Islamabad. They were acclimatized to experimental conditions in laboratory for 2 weeks in V-shaped tanks. If it twice daily to appearance deviation on the basal diet used in subsequent digestibility study. Aeration was provided round the clock to all the tanks through capillary system. Water quality variables such as water temperature, pH, and dissolved oxygen were monitored. Processing of Moringa oleifera leaf meal. Moringa oleifera leaf meal were obtained from southern Punjab. They were soaked overnight in attempt to remove saponins and other water-soluble anti-nutritional factors. They were placed on a wide mesh to drain excess water and then spread on plastic sheets to dry under shade to avoid loss of vitamins through photodynamic damage or oxidation. They were threshed from stalks to reduce the fiber content in the meal. Then they were grinded into a fine powder using a hammer mill and stored in plastic bags at room temperature. This is the figure of bengali leaf meal. This table shows the ingredients composition of test diets. Feed ingredients and experimental diets. Feed ingredients were analyzed for chemical composition. The reference diet was prepared to supply adequate levels of required nutrients for normal fish growth. Chromic oxide was used as an insert marker. Formulation of wellers, the feed ingredients finely ground into pass through 0.5 mm sieve size. All ingredients were mixed in a mixture for 5 minutes and fish oil gradually added thereafter. 10 to 15% water added to prepare suitable dough. They were processed by a converting machine to make feed pellets. This is the feeding plate and mesh. And this figure shows wall soap tanks. This is the fecal collection tube. Pieces were collected from the fecal collection tube. These are the following formulas through which both was studied: weight gain percentage, specific growth rate, FCR, feed conversion ratio. Nutrient digestibility was calculated through this formula. Chemical analysis of feed and feces. Root protein was determined by microjoule operators. Petroleum ether extraction method was used to detect root fat through Stochastic operators. Gross energy was determined by oxygen-formed calorimeter. Dyes and feces were digested in boiling nitric acid and perchloric acid mixture 2:1. Through which minerals were estimated by atomic absorption spectrophotometer. Phosphorus was estimated by spectrophotometer and sodium and potassium was determined by flame photometer. Statistical analysis collectively data of growth, performance, nutrients, and mineral digestibility of test diets was subjected to one-way analysis of variance. The differences among frequencies were compared and considered significant and probability less than 0.05. The post-stat tomato package was used for statistical analysis. Results of the current study concludes that the maximum nutrient digestibility were found maximum such as 70.74% for crude protein, 79.12% for crude fat, and 61.07% for gross energy. At 10% MOLM replacement level when compared to control diet and other MOLM replacement diets. The maximum weight gain percentage 251%, minimum FCR 1.35, and STR specific growth rate 1.39 values were found at 10% MOLM replacement level and showed that fish was in more healthy condition in comparison with control and other test diets. The majority of minerals like calcium, sodium, potassium, phosphorus, ferric, and aluminium from MOLM based diets were found significantly higher in fish body at 10% replacement level than the control diet and other test diets. This table shows the growth parameters of Lebuorhita fingerlings spread in MOLM based test diets. This table shows the means of apparent nutrient digestibility coefficient percentage of Lebuorhita fingerlings spent on MOLM based test diets. And Table number 4 shows separate mineral absorption of lipoprotein spread on MOLM-based diet and control diet. Conclusion: it was concluded that the 10% replacement level of fish meal with MOLM is optimum, which release adequate amount of chelated nutrients for maximum growth performance of the Vivahita fingerling. It also reduces the discharge of nutrients into the water, resulting in environment-friendly and cost-effective fish culture. These are the following references which I use in my research topic. Thank you for your attention.
Thank you very much, Ms. Satiyat Wasun. It was really nice talk. The last presenter of this session is Ms. Fawzia Bukhari from Kohart University of Science and Technology. She will present her work on the topic of isolation and characterization of gelatin from scales of Labiorrheta. Please, Ms. Fawzia Bukhari.
My name is Fawzia Bukhari. I am a MPhil student, Department of Zoology, Kohart University of Science and Technology, under the supervision of Dr. Farmanullah Dawar. My research topic name is Isolation and Characterization of Gelatin from Scales of Labiorrheta. The presentation outline of my research topic is Introduction, Gap Study, Objectives, Material and Methods, Results, Conclusions, Recommendations, and Differences. Gelatin is the abundant source of protein in the animal kingdom and are favorable for fin formation, biodegradability, biocompatibility, and softness. Gelatin is commonly obtained through hydrolysis of collagen and has been widely used in food industries such as meat products, cakes, ice creams, beers, fruit juices as a gelling agent, stabilizer, emulsifier, thickener because of its unique functional properties. Based on the source from which it is derived, gelatin are classified as bovine gelatin, porcine gelatin, and fish gelatin. The most widely used form of gelatin are bovine and porcine gelatin because they are easily available and have low cost. Although skin and bones of mammals are traditional sources of gelatin production, they are unacceptable in some cases because of religious constraints and high risk of zoonotic diseases. Fish skins and scales have recently gained increased interest as an alternative sources to mammalian gelatin with no religious issues and have low zoonotic effect. Hence, there is immense interest that can be potentially used as an alternative to mammalian gelatin. There are two types of gelatin with distinct properties, including type A gelatin produced by acid hydrolysis and type B gelatin produced by base hydrolysis. Gap study. Fish byproduct, if they are not properly utilized and discarded as waste, can pollute the environment. Therefore, for extraction of gelatin from fish processing, byproduct can effectively overcome environmental pollution and will impart value to the otherwise waste product. Therefore, the present study is designed with an aim at the extraction and biochemical characterization of gelatin from scales of Labiorrheta. Objectives: to synthesize and characterize gelatin from scales of Labiorrheta, to compare physiochemical properties of the potential fish gelatin with commercially available gelatin. Methodology. First, I collected fishes from Kohart, and then I removed scales from fishes with hands zipped in polyethylene bags and quickly transported to the laboratory. I took 500 grams of fish scales, washed and dried it, and then I prepared solution of sodium hydroxide and treated fish scales with 0.1 molar solution of sodium hydroxide to remove non-collagenous and unnecessary proteins from fish scales. I changed solution after every 3 hours. After treating fish scales with sodium hydroxide, I washed fish scales with distilled water until they were neutral or slightly acidic. After that, I prepared 0.4 molar solution of hydrochloric acid and treated fish scales with 0.4 molar solution of HCl for 90 minutes for demineralization. After that, the treated fish scales with HCl. I treated fish scales with HCl solution, washed it with distilled water so that it become weakly acidic. After that, distilled water was added to the fish scales and heated in autoclave at 121 degrees centigrade for 2 hours. Then I filtered resulting solution to the Wortsman filter paper so through this method, I isolated gelatin from fish scales. There are various methods to which characterization of gelatin takes place. UV visible analysis shows detection of gelatin. SEM analysis shows morphology of gelatin. XRD analysis shows nature and crystalline size of gelatin. And FTIR analysis shows functional group identification. UV visible spectrophotometer was used to characterize in the range of wavelength 200 to 400. My peak also came between 200 to 400, which confirmed distinctive structure of gelatin. This is the UV visible analysis of commercial gelatin, isolated gelatin, and commercially available gelatin. FTIR spectra was recorded to detect the commercial and structural nature of gelatin powder. This is the FTIR of fish scale gelatin and commercially available gelatin. I have also compared physiochemical properties of potential fish gelatin and commercially available gelatin, and there is a little difference between potential fish gelatin and commercially available gelatin. Conclusion. This study reveals the potential of laboratory scale as raw material for gelatin production, giving relatively high yield. Gelatin was characterized using UV visible spectroscopy, FTIR, SEM, and XRD methods. Morphological investigation showed that this polymer exhibits microsporous morphology. FTIR spectrum indicates chemical bond formation of gelatin, where XRD analysis revealed the crystalline nature of it. And we also compared physiochemical properties of the laboratory fish scale gelatin and commercially available gelatin, and there is a little difference between their physiochemical properties. Acknowledgement. I am highly thankful to Dr. Famala Dawar, Assistant Professor, Department of Zoology, CUST, and I am also thankful to Dr. Saeed Ahmed Khan, Associate Professor, Department of Pharmacy, CUST. Thanks to all faculty members and lab fellows and friends. Thank you.
Thank you very much for your attention. I hope you all enjoyed these scientific talks. Next session will start after a while. Ms. Sabarias will be the moderator of that session, which comprises from the talks of COVID-19, entomology, and pest management. Thank you very much. Stay tuned. Allah Hafiz.